Cells in the microscope. We'll start with the microscope. So, it was such a huge deal. Inventing a microscope was such a massive game changer for biology. It was a hugely important moment in the history of science, right? So without it and before it, remember, people had all kinds of strange ideas about where living things came from. There was this whole idea, going back in history that living things could just spontaneously just appear out of nothing, out of nowhere. Or out of nowhere, what I mean is, out of ponds, things just appeared spontaneously, just came into being, like, like magic. They thought that you could. I saw a great one from a fairly famous scientist from the history of science, but going back a long time, and he contributed loads of really important things to the history of science. Like, we'll meet him when we talk about photosynthesis, for example, later on. But he also had this strange notion that you could take a jar and put dirty sort of linen into it and leave the jar for a couple, and grain, right, into this jar and leave it there. And there was a way of making mice that if you came back a couple of days later and you emptied out the jar, there'd be mice in it. And his conclusion was that you could make mice from stuffing dirty laundry into the jar along with some grain. So it's sort of incredible to think that people just, but that was the understanding of today, So only because of microscopes then things moved on. We realized that what we call cell theory, in other words, things don't spontaneously rise out of nowhere. That all living things are composed of cells, and that all cells arise from other cells. That's cell theory. That's what we know as the continuity of life, right? This whole continuity theme we were talking about in our introductory lesson. So there's no such thing as spontaneous generation. And microscopes were a huge part of figuring that out. So the best early microscopes, this is a great story, right? This guy, Antony van Leeuwenhoek, he's a Dutch textile merchant. So he's fairly well off, and he's got this hobby of his, and he likes to make lenses. Now, he's a textile merchant, so it makes sense that he wants to make the best magnifying glasses possible, right? Because he wants to be able to check the quality of the fibers and do all that kind of stuff. So all that there were these handheld lenses. That was what he was using. That's what all textile merchants were using. That's what most people were using if they wanted to magnify anything. There were no microscopes, in general sort of circulation. There were some serious fancy microscopes being made by Thomas Hooke over in the UK, who was the king's scientist, but he had plenty of money to kind of build these fancy compound microscopes. Over in Holland, Antony van Leeuwenhoek was building these single lens magnifiers, and he was getting really good at it. So his whole thing, and he made hundreds of these things, right? His hobby was to grind these lenses, and he quickly realized that if you wanted really good magnification, you had to make them practically spherical, which meant that making them really small was was the way to go, because otherwise you'd end up with these big spheres, and they were hard to make. So he was making, out of just shards of glass, he was grinding them into these spheres and mounting them in these brass kind of things. They're only about this big, right? So the little handheld things that you hold up to your eye like that, and you put the specimen, I don't know if you can see it, but you can put the specimen on a little spike that's just in front of the lens. The lens is that tiny small dot thing on the left hand side of it. I don't know, I'm pointing to it now if you can see it. Okay, so he was able to see all kinds of stuff that nobody had ever seen before. He saw the first bacteria, saw the first sperm cells, saw the first algae that were in all kinds of protists, actually, that were in ponds. Nobody had ever seen any of these things before. So when he drew them all, and eventually he sent his drawings all to the Royal Society, to Robert Hooke, the king's scientist, all these fancy professional scientists, most of them lords and gentry, and they didn't believe it. They looked at all these pictures, and Robert Hooke looked at them and went, "No, he's making stuff up." So it took a bit of convincing for Van Leeuwenhoek was not impressed that they didn't believe him. So he started adding more and more diagrams and explaining all of what he'd done, and sent them back to them. And so eventually Robert Hooke went to his microscope, and he started to try and beef it up to try and see if he could repeat what Leeuwenhoek had found, and eventually did, embarrassingly for the king scientist, found that Leeuwenhoek hadn't found all, discovered all these things. So here's Hooke's microscope. You can see on the right hand side, it's this very beautiful thing with multiple lenses in it. What Hooke had to do in order to be able to see what Leeuwenhoek had noticed, he had to add loads and loads of light. Bear in mind, there was no switching on the light then. So he had these magnifiers, which were like focusing sunlight basically onto the specimen so that he could eventually see what Leeuwenhoek was seeing with his simple lens microscopes. The lens that the microscope that you use is kind of like Hooke's microscope, right? It's a compound microscope, two lenses. You've got a lens in the eyepiece, which magnifies, doesn't magnify much, usually 10 or 15 times, 10x or 15x, and you'll see it written on the eyepiece. So if you're asked what the function of the eyepiece is, it's not just for looking through, right? It's for magnifying. But it's in conjunction or in combination with the objective lens. So you can see the objective lens is the one that's on, you have a choice, and they rotate on what's called a nose piece. So you've got those objective lenses, and again, they would have the magnification written on them. To get the overall magnification, you multiply the magnification on the eyepiece with the magnification on the objective lens, and you get your total mag. So times 10 on the eyepiece, and say times 40 on the objective lens, that gives you an overall magnification of 400. Words like magnification are really important. If you're asked what the function of the lens is, you've got to say magnify. You can't say zoom, right? So you've got to say magnify. It's not much, you can say makes the image larger, I suppose. Actually, you'll get away with that. If you're asked what the focus knob is for, and there's usually two, so there's a smaller one and a bigger one. The focus knob, you can't say it's for focusing because you're not explaining what focusing is. So you've got to say that it sharpens the image, right? It brings the image in, it brings the image into, a sharp view or something like that. But sharpening the image is a good way to go. One does it roughly, that's the coarse one, and one is for, more precise tuning. You're trying to find other words than using fine and coarse and focus and all that. That's a lot of what biology ends up being, is just using a different word to describe what you're trying to describe. Then the stage is the bit that you put your specimen on, right? And usually what the focus knobs do is they raise and lower the stage closer and further away from the objective lens. That's the way most of these microscopes work. And then in the stage, there's a hole, and you need to position your specimen usually on a glass slide, directly over the hole. And that's what the objective lens is pointing at. Now, what's the point of the hole? These microscopes rely on light coming up through the specimen from underneath, through the hole, through the specimen, through the hole in the stage, through the specimen. That's why it's a glass slide, clear glass slide. And then up into the objective lens so that we can get an image, right? For that to work, the images, or rather the specimens that we're using, have to be very, very thin, right? That makes sense, because otherwise the light can't get through them. So that's for these kind of, there are other kind of microscopes, dissection microscopes and other thing, where the light is shown from above rather than from underneath, and there's no, but that's a different thing. All the microscopes we use, and the ones for the syllabus, are these light microscopes, which light comes up through the sample. Okay, so they can do maximum, usually 1,000 times, maximum 1,500 times magnification with a light microscope. That's the biggest you can kind of, or the most you can magnify the object by with a light microscope. So that means that there are certain things you can't see. Then you can see, these are the things that you can see with the light microscope. Cytoplasm, obviously you can see where it is anyway. You're not looking at much. It's just kind of, you can see the nucleus fairly prominently. Depending on the stage of cell division it's at, you can see chromosomes, you can see the cell wall of plant cells, you can see chloroplasts in plant cells. We don't see them because we look at onion cells, right? But if you were to say, look at pondweed under the microscope, the one that if you used pondweed for doing photosynthesis experiments where you're counting the bubbles, if you remember that experiment, if you've done it, if you took a sliver of one of those leaves and you put it under a microscope, you'd see chloroplasts moving around inside the cell. You can see the plant vacuole. Again, we don't see a plant vacuole, the central vacuole, in onion cells, but in a lot of plant cells, you'd see vacuole fairly clearly. If you want to see other stuff, the other cell organelles, you need to go up a level. You need to upgrade to the electron microscope. There's a few different types of these. We don't need to worry too much about the different types, scanning ones and transmission ones. Anyway, the electron microscope, it's rather than a beam of light that's going, it's a beam of electrons that's shone through a specimen. Again, you know, very thin. And then the image isn't collected. You don't look at the image because you can't see a beam of electrons. It's collected like old-fashioned TVs. If you remember old-fashioned TVs before they were flat-screened, probably don't. But if you did, old T's would have been some sort of weird contraption out the back, the cone-shaped sort of. That's basically a collector for streams of electrons, beams of electrons. So TVs work in a similar way, and you get this image then that, you know, can be turned into like a visual image for us to look at. So what you can see with those, and you can go up to 1 million magnification with that. And what you can see is you can see cell membranes, which you can't see otherwise. You can see where the cell membrane is, but you're not actually looking at the cell membrane. You're just looking at the light refracting off the edge of a container of fluid. You know, that's really what you're looking at. Or maybe a cell wall, but you can't see the cell. Are the electrons in electron microscope? That's a good question here. Are the electrons in electron microscope beta radiation and I'm sorry, and are they cancer causing? I don't know. It's a really good question. I don't think so. I don't think so. They're not alpha or beta radiation as far as I can. No, they're not. But the other question is, are they in any way dangerous? I don't think so. But I'll come back to you now. It's a great question. I've never thought of it before. You can see cell membranes with the EMs. You can see ribosomes. They're too small to see with the light microscope. You can see the nuclear membrane. You know, you can actually see the membrane itself, and you can see mitochondria. Mitochondria are much smaller than chloroplasts, say, so you can't see mitochondria with an electron microscope, with the light microscope, whereas you can see chloroplasts, right? And obviously you can see all the other stuff. You can see everything with an electron microscope, basically. Okay, quick question, have a go. What's the function for the part labeled A? Flash your answers into the direct message there if you have an answer for what A is. The function, sorry, not the name, but the function of it. What's it for? What are we going to say? Oh, very good. Excellent, good. Adjustment, I like that word as well. It's coarse adjustment to the sharpness of the image or something along those lines. A lot of you are saying rough sharpening, that's perfect. Sharpening of the image, all that's really, really good. Look at the second question. Have a look at it there and see if you might need a calculator. See if you can figure this out. This was a bit of a curveball when it was asked, but there's no reason why we shouldn't be able to, and seeing it was asked, we're not going to be caught out next time, right? So the eyepiece lens E has a 10 times magnification. The objective lens has 40x written on it. So what does that tell us? One, about the overall magnification, and what does it tell us about the actual diameter of the cell? That when you look at it down the microscope, it has an apparent size of 0.8 millimeters. What's the actual size then? Very good, got a correct answer straight in there already. Very good. So I'll tell you how to get at the answer, but have a go, right? You've got nothing to lose. Have a go. What would you do with those numbers? Yeah, that makes sense. Okay, very good. Okay, so one thing you got to remember is this cell obviously must be many times, in fact 400 times smaller than 0.8 millimeters, right? Because it's microscopic. So it looks like it's 0.8 millimeters, nearly a centimeter big, but in actuality, in reality, it's 400 times smaller than that. So how does that work for your numbers, right? If the size you're given is 0.8, but it's actually 400 times smaller. How do you do 400 times smaller? You divide 0.8 by 400. So the actual answer is 0.002 millimeters. That's the size of the cell. So good work, those of you who you know, it's not straightforward, right? It's not obvious, but now hopefully we get a better handle on it. So the magnification, if it's 400 times, that means that whatever you're looking at, the apparent size is actually 400 times smaller than that. Okay, So our answer is, folks, in the end, our answer is 0.002. I think 1/500th is actually what that is. 0.002. 2,000ths, 1/500 it is. So well, that's a correct answer as well. So either of those is perfectly good. Okay, let's move on. Probably know the difference in these two, but it's really important. I love asking this. It's going to seems to appear in every paper somewhere. You know, do you know the difference between a eukaryote and a prokaryote? So the difference between them is largely to do with the nucleus. Is it there? Is it not? Eukaryotes have a nucleus, prokaryotes don't. But there are other things. So prokaryotes don't have these things, what we call membrane-bound organelles. They don't have any of them. What are the membrane-bound organelles? Well, the nucleus is a membrane-bound, it's contained within a membrane. Mitochondria are membrane-bound organelles. They've got they're bound by a membrane, right? They're contained within a membrane. Chloroplast, similarly. So none of these things are present in prokaryotes, right? Whereas they are present in eukaryotes. And that's the main distinction. All of the prokaryotes are sorry, let me just get my participants up. There's somebody trying to bang on the door. If you get dropped, I need to make sure that I can add you back in again. So all the prokaryotes are contained within the kingdom Monera, right? They're all bacteria, and all bacteria are prokaryotic, right? So no bacteria are eukaryotic. But the flip side of that is all of the eukaryotes are in every other kingdom except bacteria, and every other kingdom is populated by only eukaryotic organisms. Okay. So let's see. We've got in the eukaryotic cell, the typical eukaryotic cells we talk about. These are not the only eukaryotic cells. We know that there's fungi. Fungal cells we'll look at later. They're kind of strange, right? Especially those hyphae. They're really kind of well, we'll get to it. Prokaryote, bacterial cells we'll look at when we get to bacteria. They're, have different things going on as well. And of course, cells like amoeba largely look like animal cells, to be honest. But then there's other protists that won't look like that. Other protists will look like, more like plant cells. So within animal cells, basically what you're looking at is they contain all of the things, plant cells rather, contain all of the cell organelles that you find in an animal cell, but plant cells have got other organelles as well. Right? So chief among those is chloroplasts, obviously. Animal cells don't tend to have much by way of vacuoles. They either have none. If they have any, they're small. And so otherwise, they're kind of the same. And obviously the cell wall. So animal cells don't have a cell wall, but plant cells aren't the only eukaryotes that have a cell wall. Right? They're not the only cells that have a cell wall. Bacteria have cell walls. Not the same stuff as a plant cell wall. Fungi have plant cell walls. Again, not made of cellulose, made of chitin, which we came across last week, but they have a cell wall. This is the kind of the view under the light microscope. You're not going to see much with an animal cell under the microscope. You're going to see a nucleus. If they have a vacuole, small vacuoles, you might see them. That's pretty much it, really. Do you see the cell membrane? Technically not really. You just see the edge of the cell where the light refracts on it. You're not seeing the actual cell membrane, right? And can you see the cytoplasm? I guess so. You're looking at something. So we'll call that the cytoplasm, because you can see the edge of the cell. You can see where the cell is. So yeah, let's say you can see the nucleus, the cell membrane, the cytoplasm, maybe a vacuole in an animal cell. In plant cells, you can go one step further. You can also see the large central vacuole if they have one, and you can see the chloroplasts. That's all you can see. And you can see the cell wall. That's all you can see in under light microscope. That's all you're ever going to be able to see with the light microscope. When it comes to the electron microscope, what we're talking about then is and you need to know this word refers to what you can see with an electron microscope. The word is ultrastructure. If they ask you what the ultrastructure of a cell is, it's it's the detailed view that you can get with an electron microscope that you can't get with a light microscope. That's what they mean by the ultrastructure of the cell. So you're expected to know that things that you can you see, for example, ribosomes in the ultrastructure? Yes, you can, because they are visible under EM. So things that you can see are details like the nucleolus, which we'll look at. It's a small compartment within the nucleus. You can actually see the membranes, right? You can see the structure of the membrane, the nuclear membrane, the cytoplasmic membrane, You can see mitochondria. You can see ribosomes. These are things that are too small to see with the light microscope.